Hello everyone, I'm Mohak, and in the talk today I'll discuss the evolution of non-homologous end joining repair pathway across bacteria over macroevolutionary timescales. DNA is prone to a number of different kinds of insults, and one of the most lethal kinds of these insults is when there is an attack on the sugar phosphate backbone that can lead to double-strand breaks in the DNA, which can cause chromosomal rearrangements and in severe cases can lead to cell death. So therefore, it becomes imperative for all organisms to invest in mechanisms that can repair such double-strand breaks. Broadly speaking, there are two kinds of repair pathways that exist. The first is called as the recombination-based repair, or RBR, where a second copy can be used as a reference or template to carry out the repair. The other pathway is when the template is not available, and in doing so, the organism prioritizes stability over accuracy. And this sort of repair is called as non-homologous end joining repair, or NHEJ. And this is what is going to be the topic of my talk today as well. Now, in terms of the conservation, what we know is that in eukaryotes, both these repair pathways are evolutionarily conserved. But surprisingly, a few studies done in the past 2 decades in few genomes have suggested that in prokaryotes, NHEJ might not be as evolutionarily conserved as recombination-based repair. And this was really fascinating for us. So we set out to ask a very simple question. What does it mean for bacteria like Escherichia coli to lack NHEJ, whereas those like Mycobacterium tuberculosis to harbor it? Now in bacteria, NHEJ is just a two-component machinery. So whenever there is a double-strand break, the first protein to be recruited at the site of damage is called Ku, which then recruits another protein called as ligase D or ligD, which consists conventionally of three domains: the polymerase, the nuclease, and the ligase domain. Now polymerase and nuclease, they do the end processing for these damaged sites and make sure that the 5' phosphate and the 3' hydroxyl moieties are available for ligase to act in the presence of ATP, which then can repair the DNA to give the DNA back. Now, in our study, we have called an organism as NHEJ proficient if it codes for both Ku and ligase D. So with that, we defined three objectives for our study. And the first question we asked was, how is NHEJ distributed across bacteria? So for that, we took a data set of around 6,000 bacterial genomes, and we did an HMM-based homolog detection. What we find is that only 22% bacteria code for NHEJ, and we could divide our organisms on the presence or absence of these two proteins into different categories. The first category was where both these proteins were absent. Second was with incomplete machinery, coding for either Ku or ligase D. And finally, coding for both Ku and ligase D. Now, if they coded for a ligase D where all the three domains were present in the same protein, we termed them as conventional NHEJ. And any other combination of these domains were termed as non-conventional NHEJ. Now, given these 22% bacteria, then we try to identify if there is any pattern associated to them. So for that, we constructed a phylogeny. What I'm showing you over here are just the representative organisms. And these are, the branches are color-coded on the basis of the phyla or the clade to which they belong. And I'm only showing you three clades, the red, green, and blue. And we overlaid the information of presence and absence of these four domains involved in NHEJ, along with the presence and absence information of RECA, which is a protein involved in homologous recombination that acted as our positive control. Now, what we see is that as compared to RECA, which is present in all these organisms, there is a patchy distribution to the domains for NHEJ. And just to give you one example, if you focus on this red clade over here, which is the proteobacterial clade, you can appreciate the sporadicity associated with the four domains involved in NHEJ. Some bacteria have all the four domains, whereas other bacteria do not have even a single domain present. Now, given this sporadicity, then we asked what evolutionary history could have presented them with such a distribution. So we tried to look at two components, the vertical gene transfer and the horizontal gene transfer. Both of these components have been known to shape bacterial genome evolution. And at the basal level, both these components are then acted upon by natural selection, genetic drift, gene flow, and so on and so forth. So first, looking at the vertical component, we did something called as ancestral state reconstruction. So for that, we use a Bayesian statistical approach. And we asked at each of these nodes and branches that what is the most likely state of NHEJ protein at that point. So when we did that, what we find is that most likely the eubacterial ancestor was devoid of NHEJ. It was independently gained across different lineages. But having said that, it was not sufficient to stabilize the trait because there were multiple independent secondary losses and gains thereafter. And just to give you one example for molecules in this case, so molecules have been known to evolve from gram-positive bacteria by reductive genome evolution processes. So if you look at this tree on the right, there was a primary gain at the ancestor of firmicutes and molecules. But at the ancestor of molecules, there was a secondary gain. Now, molecules consist of mycoplasma, phytoplasma, which are parasitic to humans, plants, respectively. So what this suggests is that NHEJ might be either dispensable or costly to be maintained in these genomes for a parasitic lifestyle. Now, to gain NHEJ, one can think of three scenarios, either directly or sequentially. What we find is from our data set that the most common kind of gain was a sequential gain where ligase D was gained first, followed by Q. In the reverse direction again, then one can think of three scenarios. So what we find from our data is that the most common kind of loss was a direct loss where both the proteins were lost at the same step. One can also ask the same question in another way. So rather than asking the most common, one can ask what is the least likely transition path for such gain and loss events across the bacterial evolution? And more importantly, why is that state avoided? So for that, we constructed a state transition diagram. And what you're seeing over here are these circles. The radius of these circles are proportional to the amount of time spent in that given state. And the width of these arrows are proportional to the number of transitions from one state to the other. And what we find is that the least amount of time was spent in the Q-only state, and the transitions to and fro Q-only state were also the rarest. In addition to that, we know from studies that Q have been shown to inhibit recombination-based repair. So what collectively this suggests is that Q could be costly to be maintained alone in the genomes. Now, having looked at the vertical component, then we looked at the horizontal gene transfer component. So on this phylogeny, this arrow would represent one such horizontal event. So we tried to ask what impact HGT or horizontal gene transfer could have on shaping NHEJ's evolutionary history. So for that, we compared the species tree and gene tree for ligase D and Q. And for this analysis, we also included archaea. So what we find is that there is a high incongruence between these two tree pairs, which suggests that horizontal gene transfers could have contributed in NHEJ evolution. We also then asked, based on certain studies, we know that in bacteria, HGTs could be infrequent among distant clades. So we tried to test this hypothesis if that is true for NHEJ as well. So for that, we compared the distance matrices for gene tree and species tree for Q, ligase D, and RECA. So as compared to our control RECA, which has a very high correlation coefficient, the correlation coefficient for Q and ligase D were around 0.2. And these were significantly different from the null distribution, which is centered at 0 over here. What this suggests is that HGTs also panned across distantly related organisms, and they were not limited to just closely related organisms. We went a step ahead, and we also tried to predict the donor-recipient associations for these transfers. So for that, we used a duplication transfer loss model-based gene tree, species tree reconciliation approach. And just to give you an example for Q transfers, we see that HGTs shaped Q transfers not just among bacterial phyla, which can be seen through these blue arrows, but they also shaped NHEJ evolution between archaea and bacteria, which are shown by green and pink arrows over here. And just to give you one specific example, if you focus on this asterisk over here, which is showing an event between archaea and the ancestor of firmicutes and actinobacteria, we did a principal component analysis for these Q sequences, and we see a distant cluster on the bottom left on the principal component 1 that consists of all the three firmicutes, archaea, and actinobacteria. That suggests that HGTs might have shaped NHEJ in these organisms specifically. Finally, we looked at the evolutionary forces that could explain the lack or presence of NHEJ among bacteria. So for that, we took two genome characteristics, genome size and growth rate, which have been known to positively or negatively impact the success for carrying out recombination-based repair under a double-strand break damage, which in turn would allow us to understand if the need for efficient DSB repair could have impacted NHEJ evolution, thereby allowing us to use genome size and growth rate as good proxies for evolutionary forces. So the first observation that we had was that organisms that have NHEJ tend to have larger genome sizes as compared to the ones that lack NHEJ. And this effect is also significant once we control for phylogeny as a confounding factor, which can be seen through the phyloanova over here. We also asked that if this is an artifact of just larger genomes coding for more genes. So for that, we did a randomization experiment where we built a random distribution for median genome sizes, and we did that for 100 iterations. And what we find is that the organisms that have NHEJ tend to have larger genome sizes even compared to this random distribution. What this means is then that one can expect a higher selection pressure of maintaining NHEJ in organisms that have larger genome sizes. We also tested for NHEJ and growth rate association. So for looking at the growth rate, we looked at rRNA copy numbers as proxies. So there has been a strong negative correlation between the generation time and rRNA copy numbers. Because rRNAs are employed in protein production, that is translation machinery, so one can then think of how rRNA copy numbers would be employed to achieve maximal growth rates. So with that, when we looked at the association, we see that organisms that have NHEJ tend to be slow growing as compared to the ones that lack NHEJ. But this significance is lost when we control for phylogeny as a confounding factor. But nevertheless, the distribution of organisms that do not have NHEJ is broader as compared to the ones that have NHEJ. And again, what this means is that organisms have a higher selection pressure of maintaining NHEJ, which have slower growth rates. We then also asked if there is a correlated evolution signal between these genome characteristics and NHEJ state. So for that, we compared two models. One model where we allowed the two states, NHEJ and genome size, to evolve independent of each other across the bacterial phylogeny. And on the other hand, we allowed them to evolve dependent on each other to give us four states across the bacterial phylogeny. And we use maximum likelihood and Bayesian approaches to do this comparison. And what we see is that there is indeed a signal for correlated evolution for genome size and NHEJ. Just to give you one example, if you focus on this tree over here, the species in black are Mycobacterium genus, and the species in white are Cornybacteria genus. So when there is a loss of NHEJ, which is shown in red over here as compared to their ancestor, there is also a corresponding decrease in genome size as compared to the ancestor, which is seen in orange over here on the tree on the right. It is suggestive of a high correlated evolution signal. We also got a signal for correlated evolution between growth rate and NHEJ. And again, giving the same example, when Cornybacteria lost NHEJ, which is shown in red on the figure on the left, there is also a corresponding increase in growth rate on the figure on the right, which is shown in blue over here, which again is suggestive of a very high correlated evolution signal in this part of the phylogeny. Finally, we looked at both these components together. So we used something called as phylogenetic logistic regression because our variables were independent variables were continuous, that is genome size and growth rate, and our dependent response variable was binary, which is NHEJ state. We tested for 3 models, and based on AIC, that is a Chi-key information criterion, we see that the best predictor or better predictor of NHEJ out of these two is genome size alone. So in summary, what I've told you today is that NHEJ we see has a patchy distribution. Multiple gains and losses could have shaped the evolutionary history of NHEJ, and how Q-only state would be costly to be maintained alone in the genome. How transfers have shaped NHEJ evolution, not just among closely related organisms, but also among distantly related bacteria and between archaea and bacteria. And finally, we see an association for NHEJ with genome size and growth rate. However, genome size is a better predictor of NHEJ state as compared to growth rate. So with that, I would like to leave you on this note that the approach that we have built in our study could be then extended to other repair pathways to understand how they have impacted bacterial genome evolution, or in turn, how bacterial genome evolution could have impacted how DNA repair systems have ended up in the extant species. In that direction, one other preliminary results that I want to share is that I'm also looking at the DNA error-prone polymerases UMU-DC and DNA-E2, which we find are over the entire phylogeny mutually exclusive in terms of their presence and absence. And there is a strong correlation with GC content, where we find that organisms that have high GC tend to have DNA-E2 as compared to the ones that have low GC, which have more UMU-DC, which also ties in well with the experimental results, which show that DNA-E2 is effective in repairing GC attacks. So with that, the pipeline that we have created in this analysis could then be tested for other repair pathways, other mechanisms to understand genome evolution in bacteria in a better way. So I would like to acknowledge my PI Ashwin for that, my co-PI Anjana, and my lab members for the support throughout. And I would also like to extend my thanks to my funding agencies, NCBS, TIFR, TDU, and Department of Biotechnology, Government of India. And last but not the least, ISMB, ECCB for giving me an opportunity to talk in this, present my work in this conference. So with that, I would like to thank you all. These are my contact IDs. Please reach out to me. And because of the time I had, I could not spend more time on other analysis that I've done on NHG evolution, which can always be found in our paper, which is now published in a peer-reviewed journal. That's the QR code for that. And I would also like to take this opportunity to say that I'm also actively looking for postdoc positions. So I'll be happy to discuss any such possibilities. So thank you.